Howdy Carp. Hello and welcome to the very first Victor Black podcast. Today's podcast, we're going to be talking about a subject that I'm very interested in. I know a great many people wish they knew more about, so I decided to invite a special guest today, Dr. Scott Stevenson, along. I first heard Scott talk about intra-individual genetic response. It must have been a few years ago. I don't know whether you actually even recall the podcast you were on at the time. You were talking, I think, about recombinant human growth hormone, and you made a couple of comments there. And to be quite blunt, I haven't had the resource to reach out to you and ask these questions. I'll be kind of waiting to start my own podcast so I can get to the bottom of this conversation. So normally, what I would do at this point in time is get Scott to introduce himself. But of course, the great irony here is most people probably know Scott and they know better than me. So I will get you, maybe. I'll get you to just, for the people that don't know who you are, because I'm sure most people would, but for those that don't, maybe you could just, briefly introduce yourself, please, Scott.
Yeah. So I kind of have a foot on either side of the fence. I've been training since I was 11 and I'm nearly 50 now. So I'm doing that continuously. First competed some well over 20 years ago now. And I just have always been a very curious person, sort of an intellectually oriented person as well. And when I got done with college, I figured I want to do something that I'm passionate about. So I went to grad school to become the world's greatest personal trainer. That was sort of like, I said that facetiously, and then I realized how much I love to teach. And I was very curious. I love learning information. I love digging in. Luckily I had some really good mentors who gave me the latitude to go in directions I wanted to. So I ended up getting a PhD and teaching, being a college professor for a little while. I have owned a gym. People can find things I've written on Elite FTS and John Meadows' Mountain Dog Training site. I co-wrote his book, The Brutality of Mountain Dog Training, with him. So John enlisted me to sort of provide some of the scientific support for what he's figured out over the course of decades, literally training himself. And let's see what else. I've put out a couple books. My Fortitude Training book, the one people know the most. And then my last one was sort of a brain dump, Be Your Own Bodybuilding Coach. And it's interesting when, you're in the Far East, and I mention this sometimes when it comes up, but that's sort of a tipping my hat to the idea of being your own Buddha and learning to sort of delve into the intrinsic inner workings of what it is to be a bodybuilder. There's so much there, and then harnessing those rewards by being the captain of your own ship, so to speak. So that's my take, is that of a teacher, someone who teaches people to fish rather than giving them fish. We're going to get lots of fish here, hopefully, but but the idea of being like, you are a phenomenal example of this, Victor. So I'm so happy you're doing a podcast, because you've excelled, in the practical world personally as a bodybuilder. You're doing great things with clients. I see it on Facebook all the time. But you also, recognize there's something to be had from the scientific literature as well. But you need to be guarded as to how literally you take that in scientific terminology, what the external validity is of the studies. Does it make any common sense? And you do exactly what I've suggested so many people do, is don't be bamboozled. Let your mind be glittered by the fact that it's a scientific study that was published in some journal. You can literally use just basic average intelligence, common sense, and get a lot from a study and see whether or not it is really something that's valid for you in the trenches or not. And that's what you do really, really as well as just about anybody I've seen. So I appreciate what you're doing too. You're helping. We're on the same team and trying to blend these things together and make sense of them.
Yeah, I agree. Thank you. And by the way, if anyone doesn't have a copy of Scott's book, Be Your Own Bodybuilding Coach, I highly, you certainly got my buddy, so highly recommended reading. So we'll talk more about that at the end, where people can get into contact with you and look at the resources you make available. But for now, what I would like to do is maybe jump into the subject that I wanted to really get some feedback from you, ask some questions. That is on the subject of intra-individual genetic response specifically to enhancement inputs, but I think it might help the people that follow me along a little bit if we just start just one notch before that, one increment before that, and just briefly talk about the idea of intra-individual genetic response to all training inputs. Okay.
Mm-hmm.
So just by way of introduction to that, if you can give me just a few minutes latitude, and I'm going to start pushing the questions at you.
Sure.
Just explain to people that are listening, really, if you pull out any dose response study that's been done, in an applied science perspective about training inputs, what you will find is typically they use more than one individual for a reason. They gather together a group of individuals, classically, probably, somewhere in the vicinity of 20, 30, 50 people is normal for most of those small studies. And whenever you look at the results of those studies, you'll see a range of genetic responses to whatever input is being considered, okay? Mm-hmm. And I say that because I think a lot of people don't really understand that, there are going to be outliers to any training input. It doesn't really matter what it is, whether it's natural training, whether it's enhanced training, whether it's a nutritional study. They're going to be people that fit into the middle of the curve that are typical responders, and there's going to be what I describe as hyper responders, and I don't know whether poor responders is the technically correct term, but I if we could use that term just for that thing. So I just wanted to show a couple of graphics here for people that are watching. So this first, can everybody see? Let me see. Yes, okay, great. We're going first show, so forgive me for the technical thing. This is actually a study that specifically looks at the intra-individual variability of adaptation to progressive resistance training. And the reason I wanted to show you this is you can see here, unlike most studies, this actually shows the range of individual responses in terms of changes in cross-sectional area as a consequence of this adaptation. And you can see the bars that represent each individual response. And there are outliers both at the poor responder and the hyper responder perspective. And a lot of people, tend to look at studies and say, well, there's a number, there's a marker, there's a response, but they don't appreciate what they're typically looking at is the average response. And that, of course, would be this marker over in the far right-hand corner, the benchmark, at least when you're starting out, saying that's the typical response from guys. So just show you another couple of quick slides, and then I'm going to get to Scott. This is the graphical representation of individuals, again, in a natural training study where they were looking at response from leg training. And the reason I show you this is really this guy up here. So there's an outlier here amongst these individuals that had just a ridiculous response to the same training inputs as everyone here. So
Victor, I want to interrupt you there just for a second. I use that. Right in. That's from I think the Baman et al. Study, or
correct.
Yeah, and I use that in one of my talks. And that point I actually have on a PowerPoint slide. And it starts flashing with an arrow that says Jordan Peters, Jordan Peters, Jordan Peters.
Yeah, mine says everybody hates this person.
Yeah, I couldn't read that, but yes. Yep.
So I guess the point of the slide really here is to say that, look, a degree of intra-individual genetic response occurs even with natural individuals, even with natural training, trained individuals, non-trained individuals. There's always going to be a range of responses that range from typical to not responsive or poor responders to hyper responders. So I'm hoping that we're on the same page. That's pretty typical, right, Scott?
Absolutely. I have the talk that I mentioned before. It's entitled "Why You Don't Look Like a Pro," and then the subtitle. And this is something I think that it's a difficult thing for people to relate to this idea when it comes to bodybuilding, because it appeals to many people because it's something that is your progress is a function of your willpower, of your desire, your discipline, and that of course engages the ego to a great degree, and body image is sort of interwoven there. This is an expression of who I am. I'm walking around in this vehicle, so what you're able to accomplish is always on display. But I also, then I ask people, like the subtitle of the talk is something like why you're not playing in the NBA, why you're not an NFL superstar, why you're not working for NASA as a rocket scientist, why you're not a musical prodigy. Like, go through any endeavor that people do, and there's people that are extraordinary at that, and people don't walk around like for decades, as some bodybuilders might, saying, why am I not running the 100-meter dash in the Olympics? Or why am I not playing in the NBA? They accept that. They accept this variability, which is, it's ubiquitous everywhere. We see it just across society. You just go to a mall here in the States and you'll see people with our epidemic of obesity. They're gigantic, and you'll see people that are just rail thin. So that's a normal, actually, you can even take it a step back, and that's a normal biological phenomenon that fits with the idea of Darwinian pressure and selection.
Absolutely.
Yeah.
It's why we are who we are, right?
Yeah. Well, it's good. Like, if something comes along that wipes out the species, and every member of that species is genetically identical, they're gone. But maybe some of them may have resistance. If it's a virus, a microbe of some sort, then those will be the survivors, and those are the traits that are carried on into the future, in future generations. So it's nice to have some variety so that you can weather what might come, and then evolution can wind its path for us. So it's important. It's a basic idea of biology.
Correct. Why we are who we are. Exactly. Yeah. Cool. So I guess if we can then start pulling on that thread and say, well, if that's true of natural training, if we would expect to see, typical responders, hyper responders, poor responders, whatever classifications people want to use, then it only makes rational sense, just purely rational, that when we introduce enhancement tools, that we would very likely see the same type of thing, where we have a range of response, where we have a bell curve where most people fit into the mean, but there are always going to be individuals that are outliers to that average. Yeah. So what I wanted to try and get down to today was this idea of, so what are the underlying mechanisms that result in the variation, not just from a trivial point of view, in other words, like, for the sake of knowing, but is there anything that we could do? Is there any validity to the strategies that might potentially help us to improve our, individual unique genetic response to those inputs? And I have a half a dozen examples that I'll wheel out at some point and flash them past you and ask you whether you feel there's any validity to them. But that's really the direction I would like to go, in saying, okay, so it's a little bit of a hello, Captain Obvious statement that says this occurs, hello.
Right.
I would like to try and explore the underlying mechanisms behind that, and perhaps I can present a couple of ideas that I believe why that is true, and you can help guide me and educate me there. And then we can talk about, well, if that is true, then is there validity to the strategies that would look at that? Now, maybe I'll just throw something straight out the cuff there to give an example of that. I think it's fair to say that your degree of insulin sensitivity is going to have a fairly profound effect on how you respond to the use of exogenous insulin. Okay. Absolutely. And I think it's probably pretty easy for people to connect to the fact that there is a great many things that we can do in terms of behaviors and tools to improve our insulin sensitivity. So even though there is a genetic component to insulin sensitivity, we can, to some degree, manipulate that through other tools or other behaviors, so that when we bring an enhancement tool like insulin to the table, it will work for us as genetically unique individuals in a more profound way than it would if we didn't use those strategies, if that makes sense, the framework here. But I think probably the easiest place to start is, so we just start with testosterone, like androgens, because I think not everybody uses insulin, and rightly so. So maybe if we start with the thing that most people watching this would have in common, which is the application of exogenous androgens, whether that's at physiological or super physiological doses. So I just show another slide here. This is a slide that people who follow me are probably freaking sick of, in 2003, that shows the application of 200 milligrams of testosterone enanthate to 33 individual men, and the pharmacokinetics profile of testosterone and the metabolites, DHT and estradiol. But the main point is that this black line that we're looking at is the average response, but the red markers that I pointed out is the range of individual response. So I guess my first question, Scott, is it possible that you could explain to me and the people listening, so why would we see that range of individual response to something as simple as testosterone over a relatively small group of individuals like 33 men? What would be the underlying cause of that variation?
So we'll take a step back here, and when we're talking about pharmacology, you can sort of divide that into two branches. There's the pharmacokinetics, which is what the body does to the drug, how it's metabolized, and the pharmacodynamics, which is what the drug is doing to the body. So what's its action, mechanism of action, so to speak. And we're talking there about those testosterone levels in the blood, and that upper left figure you had there are actually the free testosterone levels. So in this case, it was probably testosterone enanthate.
Correct.
That was injected intramuscularly, pretty standard way of doing this. All sorts of novel preparations now, which are going to probably show different profiles as far as this goes. But what happens when someone does an injection like that, the bolus stays in the muscle, and because the reason for having that enanthate fatty acid on there is to make it lipophilic so that it will stay in that depot in the muscle and slowly be released. As testosterone enanthate, it's not going to be able to do anything as far as binding to the androgen receptor and evoking the actions of testosterone. It passes through the liver first, and then there's an enzyme, it's a phosphodiesterase 7B, actually, there's one that breaks away that enanthate. And there's a study, and I can pass these along for your listeners.
I actually had the study pulled aside if you're interested in
okay.
Let me just throw it up so you could talk to it as
Sure.
we throw it up. This is the study I think you're referring to, right? Sorry, hang on a sec. I'm gonna get this up. Study you're referring to? The bioavailability of testosterone and enanthate dependent upon the phosphodiesterase enzyme gene?
Yep, 7B, yep.
Yeah, correct. Okay.
Mm-hmm.
Cool. Sorry, I'll cut you off. So that's the study.
Yeah, no, it's okay. So that's the one, and there's two. If I believe there's two versions of the gene that they examine there, or two. You can be heterozygous or homozygous for the gene, but basically across the two ends of or two versions of the gene, the activity of that enzyme will change, and that will dictate the extent to which blood levels of the free testosterone are bound to serum hormone binding globulin or albumin you're going to see. So your total testosterone levels as testosterone bound to those proteins in the blood is going to be a function of the activity of that particular enzyme in the liver. And the difference is substantial. It's the area under the curve. So we can think of this as the extent to which the blood levels of the testosterone are presenting that testosterone to the cells. Area under the curve is sort of used as a surrogate for to what extent at least bloodborne metabolites or drugs are able to impact the target tissues where they're going to act. The ranges, it's a 60% difference. So this is substantial. With one person, let's say, who has the less advantageous version of that gene, injects 200 milligrams of testosterone, and they get a certain area under the curve and a profile like you showed in that figure. Someone who has the advantageous in terms of getting higher, larger area under the curve from that 200 milligrams would effectively get what that other unlucky SOB would only get from 320 milligrams, a 60% advantage. It looks like, yeah, that's the figure. They've got them grouped into the two versions of the genes there. Yeah, so that's an important one right there. Mm-hmm. That's just one piece of the puzzle that dictates to what extent the testosterone is able to do its job by being free testosterone or bound to proteins in the blood. Mm-hmm. So that's a 60% advantage. That makes, you can think of that in various ways, but that makes someone who's going to use 500 milligrams. This would actually go for probably, this is what I haven't found, other esterified hormones too. So I've seen that in the context of nandrolone, but I haven't seen it in the nandrolone deca, but I haven't seen it in the, whether that applies to propionate. I would presume so. So that's our first advantage there. And
can I just interrupt you there? Would that be the same gene that was responsible for the the cleaving of all of the different esters, or?
That's what I don't know. I've looked and dug into that, and I wasn't able to find
I can't find the answer myself. So,
yeah.
Okay, so let me clarify. So we know that that particular gene is responsible for the efficacy with which you cleave that ester from the master hormone.
Mm-hmm.
And we believe that it's also true on nandrolone decoinate, but we don't have confirmation that is true of all of the different esters, propionate, cyprinate, and antate. It's plausible that it's the same gene, but it might be a different gene. Is that right?
It's possible, yes. It's possible.
Cool. Right.
I mean, for instance, go ahead.
Sorry, I answer your question, sorry.
Oh, I mean, I was going to say we know, or the suspicion too, and you'll have a hard time finding things here, is that when people talk about phase 1 detoxification of androgens, androgens are all fairly similar chemically. It's like if you're looking across the spectrum of automobiles, you've got convertibles and you've got trucks and you've got sportsmobiles, or you've got sports cars. And so all the androgens look like sports cars. So you would treat them all the same. It's the same P450 enzyme, 3A4, I believe, that is involved there. So as best I can find. So it would make sense. A lot of times nature is pretty good in economically using the same gene. Actually, many genes get used for a multitude of things. So it would make sense, but I can't say for sure. And the other thing that's interesting there is you see when in those two groupings, I think in that figure, is that the blood levels are a function of the release from the liver as well as clearance from the bloodstream. So there's going to be some variability, and you see that, like, it wasn't like we had two discrete bands, so to speak, of people with the different genotypes for that particular enzyme. There's still some variability there because the cleaving is only one part of this picture. It's a very complex one.
I understood. Yeah, correct. So if we just constrain the conversation to that, as you said, very simplistic, one element of the entire picture, what would your response be towards the discussion that you will sometimes hear that different people respond better to different esterized versions of drugs? Like, people say, well, I like propionate, I like cyprinate and antate. Would you say that it's plausible there is a underlying valid reason for that? They seem to respond better to various esterized formats, or that does or does not make sense, from a mechanistic point of view?
Yeah, well, there's a couple ways to make sense of that. This is a perception someone has, so it's
correct. It's a belief.
Yeah, it's a belief to some degree. It's what they sense in some way, shape, or form from how they feel or their results in the gym or something like that. But back to actually what I was saying is relevant to answer that question. So the blood levels are going to be a function of entry and exit. So if you think of the blood as a room, the number of people in the room is going to be a function of how many people are coming in and how fast they're actually leaving. So if you get a buildup in the blood, which is what you see in the day or so after injection like that, because the rate of entry as that esterified hormone leaves the depot, goes through the liver, and then the ester esterified fatty acid is cleaved away, you've got a rapid rate of entry which exceeds the rate of disappearance. So some people may find that you take two individuals with the same enzyme, the same gene for that phosphodiesterase 7B enzyme, and so their rate of entry is the same, but others might have, for a given molecule, nandrolone, let's say, or testosterone, might have different enzymes involved in the liver with removal as well. So there's a pacing. So now we're getting into complicated situations. We've got one door in and several possible doors out. Because one person is able to metabolize and clear the testosterone more quickly than another. And the rate at which that happens through those different doors for different individuals is a function of how fast things are going in.
Mm-hmm.
So even if the rate is going in, one door might be better in terms of keeping blood levels elevated with different esters, which are going to come in at different rates. So you've got both of those sides. Basically it's a homeostatic balance scenario. Yep. And that's just looking at clearance, like for instance in the liver. We've also got what's happening with the 5-alpha reduction of testosterone.
Correct. And
there's variability in those genes, and there's aromatization as well. So those are three pathways: aromatize, the extent to which the testosterone might be bound, and the ability of serum hormone binding globulin or albumin to bind that, and the extent to which testosterone is converted into other metabolites. So all of those have certain rates at which they'll occur depending on the genetics of the individual. And which pathway you flow down will depend on the rate of entry of free testosterone, which is a function of the gene we just talked about, the phosphodiesterase 7B gene.
Yep.
So it's very complicated, really, and there are ways in which individuals could experience, for instance, they don't like there's some research that suggests that propionate will produce lower levels of estrogen for a given weekly dose of testosterone. That may apply to some individuals, but if you look across that bell curve, which we see on everything, for some people that may not make a difference for them, for whatever reasons, and that's probably a function of the aromatase enzyme at least, and maybe even other things. The relative rate at which testosterone is simply presented to that aromatase enzyme is going to be a function of the rate at which it goes those other pathways. So individuals can have various different combinations. You start adding these up and the combinations become manifold. And that's just in the pharmacokinetic side, essentially, not on the pharmacodynamic side, where things can differ as well. So that's
let me ask a question. I'm sure there's no answer to, but I just need to ask the question. So I think it's fair to say, from what you said, we just scratched the surface with that, but already, as you said, you end up with potentially a large number of factors that influence the end outcome. It
right.
could be like many different factors. In your opinion, do you think that individuals tend to be like the individuals that have the upper hand on one of these elements, do you think that they have the upper hand across the board, or do you think it doesn't matter? There's no necessary correlation between. I always say to people, when you talk about like I'm genetically gifted or I'm genetically cursed, it's very difficult to say, because in my experience, everybody gets some good cards and some bad cards, if that makes sense, that term, Julian. And every now and again, someone comes along that just got all the good cards. That's the guy, right? And every now and again, someone comes along that just like he got hit with the unlucky stick and he's just kind of like Pee Wee Herman, doing. But do you think it makes rational sense that an individual can have all of the genetic, good cards, as it were, so that they just is that where the hyper response comes from, or do you think it's potentially like it's more complicated than that, where you have a high and a low, and that balances out to average? I know that's an impossible question to answer. I just want your opinion, basically.
I haven't seen people try to do like a full genetic spectrum and correlate to see if there's correlations there. What I do suspect is that, and let me add one more thing to make this all complicated, and this is so infrequently addressed, I just want to say it so to get it out there and get this idea in people's heads, is that the expression of those genes for those enzymes we mentioned, or those physiological mechanisms for clearing drugs, those also change over time. So
with age, or with
the progression of use of a drug. So the first time you introduce a esterified testosterone preparation, there's more than likely, and I haven't even seen this study, but I would be absolutely amazed if we don't see some change in the activity of phosphodiesterase 7B, because it's being called into action.
Yep.
So it may be such that you start, and there's probably variability here. These genes are turned on in very complicated ways. There's promoter regions, When the genes are expressed, the transcript has to be broken apart and pieced back together in certain ways. That's actually one form where you can see genetic differences, is just in the way that the gene product is put back together. There's some parts of the genes that end up in the final protein product, and then there's some parts that aren't, that are left behind. So, but those change over time. So one person might, for instance, get really, really good at clearing testosterone for whatever reason. That's their genetic proclivity. So they are someone who develops quote unquote tolerance quite rapidly. But I do think, to get back to your question there, is that if we could get some of the best, the most gifted individuals, the Kevin Lavronis, nearly, for instance, one of the things I do in that talk about why you don't look like a pro is I went and looked, it was a few years back now, at the top 10 at the Mr. Olympia, and figured out how many years it took them to get from their first contest to pro status. And it's like 2.5 years on average. And I think it was 3.5 when I included a couple of those competitors, like Sean Roden, obviously has pretty good, he had like a 7-year gap.
Yep.
When he stopped competing, I think it was when his father passed.
Yeah, it took some time, yeah.
Those individuals probably are gifted in terms of the pharmacokinetic side as well as the pharmacodynamic side. And one thing that you see sometimes in individuals, and this is the other big part of this picture in terms of how well a drug's going to work for someone, is the extent to which that drug causes toxicity.
Yes.
So a lot of individuals, a lot of pros, it seems, and the most gifted ones are just really sensitive. They don't need to use very much, and they typically don't. People.
Correct. In my experience, that's true as well. Yeah.
Yeah. I mean, it's like, what do the pros use? And like,
less than you think. Less than you think.
Yeah, but
it's the guy that comes 10th that's using the most usually, because he's the guy.
A lot of times, yeah, very much. Yeah. And so you can have, for instance, some of the best pros may be very, very sensitive. They use small amounts. I mean, Ronnie Coleman, there are a couple of pros, Ronnie Coleman, Kai perhaps, who look at what happened when they first got their card, and then they added 70 pounds of muscle afterwards. They may have been natural when they got their cards. I wouldn't doubt that.
Quite possibly, yeah.
So there's one feature to being sensitive, and then there's also the feature that there's a dose response in general to these drugs. And the way I've always sort of looked at this, and you don't see this isn't the type of thing you can study directly in humans because it's probably not ethically sound to do so, is that once toxicity gets to a certain level, it's going to slow rate of growth. You're basically going to be sort of toxed out.
See it time and time again, absolutely, yeah.
Yeah, and you see that, like, if you look in the agricultural literature, they look at, for instance, like in cattle, they look at some of the toxins that are natural defense systems of the grasses they would eat. And you can only have so much when they try to mix cattle feed and that sort of thing. You can have so much of those toxins before it makes it it slows growth noticeably. So there's various ways in which that can occur. So you may have some individual, and there are IFBB pros who have been renowned for this for various reasons, who may be very good in terms of their sensitivity to the drugs, the pharmacokinetics and pharmacodynamics, and also have a very hardy system in terms of detoxifying. So instead of getting only to, let's say, 2,000 milligrams a week before the toxicity overload gets, I'm just generically speaking here.
Sure.
The toxicity becomes so strong that they no longer get any benefit from adding gear.
Yep.
They can reach out further on that dose response curve, just looking at the impact of the drugs on anabolism, without the detrimental toxic effects coming into play.
Agreed. So that leads me to the obvious question, which is really what I would like to try and press down on. Says, well, is there any way that we could plausibly upregulate the expression of these genes?
Well, which genes? Some of them we want to upregulate, some we may not.
Yeah. Well, that's true. So can we modulate or manipulate some of these known factors to our advantage that are not commonly used practices in our tribe, if you know what I mean? So I'll give you a simple example. It is my firm opinion. I'm happy to be re-educated necessarily, but it's my firm opinion that most people incorrectly misunderstand that androgen receptors downregulate in the presence of androgens, and I have found no evidence to support that whatsoever. In fact, I found that, it's my opinion, androgen receptors upregulate in the presence of androgens, and there's no upper limit. It just keeps going, basically. Of course, at some point you reach a point that you see a blunting of growth, but that's not consequential to saturation of androgen receptors or overloading. That's another mechanism that's behind that.
Right.
So the obvious question would be, aside from the administration of androgens themselves, are there any valid supplementation solutions or pharmacological solutions to uprate androgen receptors? Just that component of to start with. Are you aware of anything that's practical, that's viable?
Well, the first thing we have to sort of recognize is that you do see adaptation to the drug's effects over time. That's you'll see that just in general, and then we also there's a sort of a phenomenon of diminishing responsiveness the larger the muscle cells get in general. So that's gonna impact regardless of whether you're talking about training or food or what have you. The muscles can only get so large, and eventually they approach a limit. So if we're looking at the androgen receptor, and you're right, the androgen receptor levels go up, and people see my dog walking in the background behind me there. That tells us that there's much more to the transactivation, the signal transduction, than just the androgen levels.
Correct. It's
what's happening post-receptor binding that's important for the actual impact of the androgens on anabolism and cellular metabolism. So here's the thing, and this is within our tribe. People know and they do this, but I think it can be something that's, there's a couple things actually that's underutilized. One, if you look at the androgen receptor itself, there's some interesting things there in that you see, generally speaking, you can go through, there's a nice article you may have found it's one of the only ones of this kind, by Saartok, Saartok. And they look at relative binding affinity of various androgens, and you see in those that are typically used as esterified injectables, pretty good binding affinity. And then you'll see in drugs like Dianabol and Winstrol, like the binding affinity is really, really low.
Yes,
it doesn't bind to the androgen receptor very well. And that's not really totally understood at this point. There is a particular study, which you may have found too, where they administered, I think, trenbolone, testosterone, and they also had methendienone, D-bol, and stanozolol, Winstrol in there. And they found that there was a dose response in terms of the transactivation that they saw. So there was a cell culture set up where they could look to see to what extent they're activating those androgen receptor genes. And they got the same maximal response from all of those androgens tested. So, and we know that you don't have to take 10 times, you don't have to take 5 grams of D-bol a week to get an effect from D-bol.
Correct.
But if you look at just the binding affinity to the androgens, that's obviously not telling the whole story. And there do seem to be now, there's the classical cytosolic androgen receptor, where the androgens, because they're lipophilic, they make their way into the cell, the dimerization, and then you have the impact on the in the nucleus, and that's something that takes a long while to take effect. Steroid hormones with that classical mechanism, it's going to take days before you start to see anything. It's just a slow process. But those androgens in the orals fall into this category, also can produce immediate effects in neurons and even in skeletal muscle. And there's probably a plasma membrane on the surface of the cell, androgen receptor, that is different than that classical genomic based one that takes a long time. That's the one we've looked at so much. So
just where you said that, could I just ask you a question about that? Sorry, I'm diverting. It's my understanding that those androgens that show a greater affinity for the membrane-based androgen receptor also are correlated to much higher levels of neurotoxicity. Would you agree with that statement?
It's interesting because those are the ones that are typically used as orals. And we know orals, because of the route of administration, are going to bring that about because they've been methylated, basically. So these are 17 alpha alkylated, and they cause liver stress. The point is is to evade the liver's ability to detoxify.
Sorry,
and
sir.
clear them.
I might have be clear. I meant neuro cognitive function, neurotoxicity.
Right. Well, the toxicity is going to be global. It's going to be in the brain
Okay.
and otherwise. Yeah. I mean, trenbolone is typically, you can get, base trenbolone, and you could use that, but trenbolone is known to increase plaque formation that's associated with Alzheimer's. So yeah, so it's not, they all have individual effects, but yeah, the thing that's interesting is you mentioned the differential binding affinity to that surface, the cell membrane androgen receptor. That's a very mysterious androgen receptor. They haven't done there's not a really steady, deep, progressive research line digging into what is done with that androgen receptor. But the thing that we can use sort of from practical, just going back to the practical realm here now, is that some people find, for instance, like in an even more global way, like some people say, I'm a peptide guy. So they do better with insulin and growth hormone as opposed to gear. And so there's genetic reasons for that, of course. And then some people don't do very well with orals. Some people, it's just like, I'll never take orals again. It's the worst thing for them. They really suffer those ill effects. So they may be someone who's not able to deal with the toxicity, but we know that we've got different mechanisms through which these androgens are all working. And there's going to be, for each of those, the thinking here now, the plasma membrane receptor and the cytosolic receptor. So there's going to be the potential that someone is more responsive to one versus the other. So that's something that someone can dig into and start playing with. And there's also the idea of figure out which do I do better with, and which can I use without suffering toxic effects. And we mentioned Latz from Pro Muscle. He was on my podcast, and you've talked with him. And on my podcast, he talked about, this was just a brilliant approach. And I wish I had thought of it myself, but he talked with a competitor who, over the course of time, had sort of categorized for himself. It was basically sort of androgenic and anabolic categorization, the typical way most people would do that. And he pieced together combinations of two androgens, which he would incrementally increase until he reached what he felt like was the law of diminishing returns was applying, and he was no longer getting anything from adding more to those doses. And he may even use them both individually. So he figured out which combinations worked for him, in which doses that worked for him. And then he started recombining things, and he came up with his own personalized system of combining different steroids in a way that allowed him to cycle on and off different combinations of steroids that suited his particular genetic predisposition for gaining from those different combinations. So some people might find, and of course, using orals long-term is probably not the best option here. And probably for some people, not really a viable option at all.
No.
But some people may find, because nowadays, I mean, you talk about the old days, guys would take 10 milligrams of D-ball and they grow like weeds. And I think people miss out on that. Maybe because if someone starts taking, let's say 30 or 40, because that's what they read they should do somewhere online, they may actually not get as much as they would have gotten out of 10, because that's so much that it's causing toxic ill effects for them. That's reducing the progress they would make from that. But finding a way to figure out, for instance, all of these things, the pharmacodynamics, which esters work better for someone, as we've talked about, and which combination of which steroids work, because they all seem to have potentially different combinations to which they bind to the cytosolic receptor versus that membrane receptor. And it's going to be a very personalized thing to some degree. It's going to be a trial and error, which is what bodybuilders have been doing now for decades.
Which really leads me to my interest in this. So my take on this is that although it's very interesting to read these studies and try to understand the underlying mechanisms, I think it's fair to say it's extremely complex, and there are certainly areas that we still don't have the full picture on, and will be probably years before we can go in and have go and get a test that tells you what to take and when.
Right.
In the meantime, really, this, again, this is why I have the great passion of trying to keep one foot in both camps, the world of the anecdote, the experience, and one camp in the camp of the evidence. Because I look at this and say, look, the only real answer I have to this question is trial and error. You have to get in there and expose yourself to these compounds and see how you as an individual respond to these compounds, and rotate through different cycle models and try different anabolic compounds and various inputs, and see how you respond to it. Would you agree that there's not really a scientific method that you can deploy here, or would you say there is a model that someone, I'll give you an example what I mean. I think if you're genetically gifted, most truly genetically people go into the gym, start training and start growing and never fucking stop. Basically, that's what happens, right? And I think that I'm really interested in learning about this for the people that come to me that just seem to struggle. Like they just don't seem to get the return on the investment that one would think that you should. And after working with them for a little while, saying, well, they are doing the right thing with nutrition and sleep hygiene and stress management and appropriate training models and things like this, and they seem to be poor responders. And I'm trying to understand better if there is a methodology or a model that one could follow to try to understand what drugs might work better with particular individuals based on whatever behaviors or whatever profile they might particularly fit. Is such a thing even practical to invest my time looking at, or you think it's just, you've got to try it and see everybody's gonna, you can't look at anything to say this is predictive of such outcome?
Well, I think you can be very scientific and meticulous in the approach that you take, like this person I mentioned that Latz had spoken with.
Documented everything, yeah.
And so there's two notions there that you can kind of keep as guiding rules, I would say. And one is that there's going to be some adaptation that occurs over time.
Yep.
So, like you said, the gifted people, they don't seem to show, they just keep growing and growing and growing and growing.
Yeah.
And that's why there's this, it's sort of a logical fallacy to look at those individuals who did not have to follow the path of the hard gainer in any way, shape, or form as ways to figure out how to look like them, because you don't have their parents. You don't have their DNA, so they're playing by a whole different set of rules. So you can't apply their playing strategies. Just go in the gym and kind of mess around and train like Paul Dillett and eat fast food and what have you, and expect to make those kinds of gains. So, but the one thing we do know is that people are going to show adaptation, and they're going to stop progressing at some point in time. And so if you take that and then the idea of get the most from the least, which I've always sort of applied in various ways, is you can try to introduce, let's say we're talking about androgens. You could, for instance, here's a way to go about it. Consider an oral and consider an injectable, and even consider one at a time. In an ideal world, you only change one input.
One variable at a time, yeah.
And this doesn't fit with the mindset of trying to get everything all at once. Yeah. So you may have to practical limitations. If you got a show on the road, in the future, you can't spend 5 years, just adding one thing at a time. But you could try, for instance, one injectable, and then over the course of maybe 3 or 4 weeks, even in an off season, use an oral, which can be used with a timing approach. Just like nutrient timing, you would only use those on the days you train, which is a nice thing because they can sometimes be pre-workouts in terms of increasing aggression during the training, and see what happens there. And you could actually do a blood work measurement. You could look and see what happens to your liver enzymes over that period of time. You want to have a washout thereafter and get back to a baseline, so you'd add something else. But someone might go 3 weeks on, 3 weeks off, and over the course of 6 months, let's say, I'm concocting this in my head right now, given what we talked about. They might try D-ball at a basic dose, or maybe an incremental dose, starting with small amounts, because the first 10 milligrams they'd be sensitive initially to that, on a background of testosterone. And then they would take 3 weeks off. They measure the blood work before and after, clean out. This is a crossover design from an experimental standpoint. And then they would go with stanozolol at an incremental dose and see what happens there. And you could write down perceptually, how do you feel? what's your perceived recovery status. I have a PRS scale that it's actually on my website that I use with people. You can look at what's happening in the gym, keeping in mind that you've got a law of diminishing returns there. You can measure your readiness to train. You can use heart rate variability, resting heart rate, sleep scores with an Aura or whatever sleep measurement devices you happen to be using, and just pay attention to those things and look for kind of a pattern. And it's very clear, like an obvious example is some people, trenbolone is like the most controversial one. And some people like, I can't sleep on tren. I feel horrible on tren. There are ways you can kind of combat that. Dr. Dean St. Martin did a really nice presentation on Fuad's podcast, which you can listen to on that. So that would be one way to look at those things. So adding incrementally doses, start with smaller doses and add as needed. Trying to adjust the dose as adaptation occurs. And then, of course, even if you, after that washout period, there's some possibility, if we consider that different orals do have somewhat different, there's some variation in the binding affinity, and they seem to work differently. I mean, no one's going to argue that Anadrol is the same as Anavar. Like those have different impacts on the body. Some people do well with one and not with others. Some people can do well with great. Some people think Anavar is the greatest thing since sliced bread.
Absolutely.
There's something going on there. And so those effects are going to slowly diminish over time. So that's the time when you're not getting anything out of the dose. You just basically have an ongoing toxicity issue, which can even get worse if your liver is continually unable to keep up with the stress of the androgen. And so then you would switch to something else. And ideally, then whatever mechanisms that are involved with the impact on water retention, fat loss, Anavar seems to have an impact on fat loss that people seem to like. Those things would be, if you've switched to Anavar, let's say, those things would be fresh and ready to go for a smaller dose of Anavar, because those mechanisms wouldn't have been in play with the previously used androgen.
So it sounds to me like what you're describing would be saying, look, our best chance is through the selection of the compounds in our already existing toolbox and trying to determine what you as a unique individual respond to, other than saying, well, I was kind of, I guess this is what I was, a bit of a fishing expedition sort of thing, saying, well, is there anything that we could do in terms of behaviors or compounds to say, look, we'll stick with these compounds and try to elicit a better response from an individual by adding something, either a behavior, a supplement, some pharmacological solution. It sounds to me like what you would recommend to those people would be to invest in well-documented personal experimentation with the usual suspects. Is that fair? That's one thing. Yes, absolutely. There are, for instance, you can. There is a study with Synephrine. It's actually in my book, where they use, and you've heard of people drinking grapefruit juice or avoiding grapefruit juice if they're taking certain drugs. It inhibits P450 enzymes. So, for instance, if you want to, you can increase the half-life of androgens by inhibiting the CYP3A4 enzyme, which grapefruit juice will do. I think both the naringin and naringenin in there will do it. I looked this up. I think I wrote it down. Yeah. Golden seal does that, as an herb that does that. Piperine from black pepper. You go above about 10 milligrams. You usually don't see anything more than that in most supplements that have included the piperine. That also has an effect. Though there are ways that you. So I'm assuming most of the literature that talks about this is not referencing anabolic steroids. It would be looking at other esteroid drugs. Would that be fair?
Well, looking at steroid drugs in general.
Yeah.
So, for instance, someone who's on a corticosteroid.
Yeah, correct. Yeah.
For inflammation, or someone who's taking a birth control as well.
Yep.
Yeah. So it seems like all the steroids, because they're all generally similar in their major structure. They got the steroid rings. They're all metabolized, as far as I can tell, by that same CYP enzyme. So you can basically make the liver less effective at clearing those in the phase 1 process. The phase 1 in the liver, for listeners who are interested, you basically, it's a process that you change the configuration of the molecule, but it also tends to produce free radical stress.
Mm-hmm.
So most of these liver supplements you see with like alpha lipoic acid and all the other components, a lot of those are helping counter free radical stress. And then thereafter, that modified version of whatever the toxin is, the xenobiotic, in this case we're talking about steroids, gets conjugated to some other compound so that it can be more easily lost and excreted from the body. So that first step of the conversion can be inhibited. You can impair that with these over-the-counter supplements. So that's the danger there, and this is why I don't go right to that, is that you can have global effects on how you're metabolizing other drugs too. So
other drugs, correct? Yeah.
Yeah, you could shoot yourself in the foot, in terms of your aromatase inhibitor. If you got a polypharmacy approach going on here, you can run into problems doing that, because it's not a very refined tool as far as that goes.
Okay.
So
I'm just looking at the clock. I already see it's a whole crap. An hour's gone already. I just spent 5 hours talking to you. Can I just want to, because I would really like to ask a couple of questions about growth hormone. Can we just jump onto that? Otherwise, I'll be sitting here talking to you for 3 hours, and I don't want to do that. So let me just go back to that. Tool that I had, so I could be a pharmacokinetic study of what you could only be described as high range recombinant human growth hormone. This is the one where they went up to 0.3 international units per kilogram. I'm sure you know the study I'm talking about. I'm curious about the massive range of response here in terms of IGF-1 levels. It just seems like beyond belief that there are individuals out there that could, be exposed to these types of dosages and yet, barely break above natural range. If we assume that 300 nanograms per milliliter is, normal range, roughly, somewhere that's lower than that, but let's just for ease that, you would think by smashing away at those levels of growth hormone that you would be expecting to see, multiplying factors at play here, and clearly you do in some individuals. I'm just curious to get your take on, the genetic response to the application of growth hormone. Now, clearly, I'm assuming there are also enzymes at play that are consequential to the thing. It's a very similar story that plays out with the applications of androgens here. But why would you say that we see such high ranges? I'm looking at this saying, okay, so we know that this is recombinant human growth hormone of pharmaceutical grade quality because of the nature of the study. And we know that everybody's getting the same correct dose because of the nature of the study. But I've always looked at this study and going, I've been shocked by the range of individual response. Do you have any general comments about that, or any insight or guidance as to why the magnitude of variation?
Yes, there's so much. So one of the things that people look at growth hormone and IGF-1 from the perspective of the somatomedin hypothesis, and that's the idea that somatotrophin, or growth hormone, is formerly known as that in a lot of the early literature. Its effects are mediated on growth to some degree by the production of IGF-1 in the liver systemically, and that's what you see during growth and development. Those were adults, so we're past that period. And so that's true. And growth hormone, as you saw there, does tend to increase IGF-1. But IGF-1 is also remarkably indicative of nutritional status, just in general, in individuals. So it's something that goes to shit in burn victims, for instance, when they're highly catabolic. And back in the day, a way that people could actually get growth hormone prescriptions was they just use an IGF-1 test. They wouldn't even measure growth hormone. And if you were dieting down and you were in a caloric deficit and you had been for a while, your IGF-1 would be in the shitter.
Trash it, correct, yeah.
It just was the case. So there's those variations too, which will come into play. There's also differences, and there's a whole slew of things. There's an article that I pulled up in preparation for this showing things like the vitamin D receptor, insulin-like growth factor binding protein 3 variations in these genes. Long known one is that there's 2 different versions of the growth hormone receptor, one with and lacking an exon 3 that changes the activity that is brought on when growth hormone binds to the receptor. So there's a ton of different genetic variations there that can impact to what extent you get the growth hormone release. So if you've got someone who comes into a study like that and they're just poorly nourished, just in general, vitamin, mineral, and or calorie deficiency, or like literally they're going to have a poor response. The body is mediating its growth and its general anabolism in the context of nutritional status. So it would be interesting to look at, for instance, what if they could do this, do a dietary recall, which doesn't tell you as much as we would hope because they're notoriously sort of off, not particularly valid.
Self-reporting as well, most of them, right?
Yeah, the self-reporting is just terrible. It's a nightmare.
Yeah.
But look at like what those if there's some correlation between the IGF-1 response and the calories per kilogram, or the calories reference that you would expect that the individual would need, or the extent to which their activity levels vary. Body fat levels make a difference. Age makes a difference. All these things vary. So IGF-1 it's such a variable thing anyway. I mean, across individuals for all these different reasons, it's responsive to so many things. It doesn't surprise me. That's a really impressive graph, though. It's all over the place.
Yes, it's wow. It's like holy crap! Like
yeah. It's barely. I mean, were those standard error bars or standard deviation? Did you?
I read them as standard deviation bars. So.
Okay. Yeah. Sometimes they put standard error bars in there. You can actually read those from the plots and see how close something was to being statistically significant or not. And if those standard error bars were like they're almost down to that baseline 0, that even though it's a substantial average rise, the width of those standard deviations.
I guess that was my interest. It's the width of the deviation that I just found shocking. I couldn't understand why, with that dose, you would see such wide variation. It seemed to be almost like no response. And this is where I was getting at. So I have read a number of studies that talk about the development of growth hormone antibodies. As part of the treatment methodology for growth hormone deficient, children and adults, and it would appear that at least a percentage of individuals over time develop antibodies to growth hormone, and one of the clinical treatment or the solutions rather to the development of these antibodies seem to be like a washout phase, clearly,
right?
But also it's recommended that you might potentially change brand, and I don't quite follow why that might have merit. I don't understand why that would be a solution, given the fact that we are talking fundamentally about a fairly standardized synthesis process today. Growth hormone is made fundamentally the same in all pharmaceutical. Why would changing the brand be part of clinical practice for managing the development of growth hormone antibodies?
So the caveat is I'm not an immunologist here, but this is a fascinating thing. So I found a study which compiled some of those studies you probably looked into, and in some studies there was no antibodies formed, and in other studies up to like 60% of the patients, these were growth hormone deficient dwarfs, children, developed antibodies. So there's, for some reason, some individuals' immune system sees the growth hormone molecule, maybe because the levels are so elevated. I don't know the reason why, but they see that as a foreign xenobiotic protein and develop an antibody to that. And when you think about, for instance, how vaccines are formed, for some molecules we can't form, for some viruses we can't form vaccines based on antibodies because they can't figure out what portion, or at least in part because they can't figure out what portion of the molecule is the one that the immune system will be highly reactive to. So you can give people a flu shot, for instance, by introducing an inactive version of the virus, but you have to give the right portion of the virus that the immune system will then respond to and say, like that's if the invader is a red car with big flashy hubcaps and, it's a pinstripe and a wing on the back or something like that. Can we put the wing into the system and then make sure that the body looks only for things with wings, or is the pinstripes the important characteristic? And there's some difference to the extent to which people do that. So antibody formation is it's individualistic, and it seems to be specific to the various features of the molecule. So just because you have 191 amino acids lined up, those have to fold into a configuration that makes the growth hormone molecule that fits into the receptor. So there can be, depending on the quality of the production process, misfolding. One of the things you'll see that people have finally kind of become attuned to is that in any growth hormone preparation, you'll have some growth hormone fragments. So there'll be some incomplete growth hormone molecules that are part of the preparation. Yep. And you can also end up, and this is a natural thing you see in the body anyway, is dimers, two growth hormone molecules stuck together. And in some cases, that may actually be functional if the part that bind on the growth hormone that binds to the receptor is still open. It's okay, as long as that dimerization doesn't change the shape of the growth hormone so that it can no longer conform to the receptor. So if you take a given preparation and it's got 3% fragments or 1% fragments in it, and those fragments are a fragment that a given individual forms an antibody response to for whatever reason, because their system does that, then they're going to form antibodies, and they're going to pick up on at least those fragments, which are fragments of the growth hormone. And potentially, if the immune system does what it's supposed to do, it recognizes anything with that fragment in it, meaning the growth hormone molecule itself, and then it binds those with the antibody, and then the immune system has its way and disposes of the growth hormone, so it can't be around. So it's considered a foreign invader, so to speak, or at least something that's foreign that shouldn't be there.
Okay, so that leads, because everything leads to an ultimate question where I'm going with this. So would you say that based on that thread, that the strategy of rotating through different brands of growth hormone over time is a valid one, or you go, look, I don't know that necessarily applies to our tribe?
Well, that's for some people, yes, and for other people, no. So for some people, for instance,
depending on whether they develop the antibodies.
Yeah, and there's also you can have glycosylation, and you can have situations where different amino acids get oxidized or in various ways that changes the shape too, and that can be a function of storage or something just in the production early on, heat exposure or what have you. So if that person is someone, and some people the antibodies will go away. The immune system will lose that. That's why they want to suggest that you get a flu shot every year to sort of maintain your antibodies in case you're someone, that loses that memory. So for some people who maybe tend to form antibodies easily, and also those antibodies also are gone relatively quickly, they might use growth hormone for 3 or 4 months and then start to find that maybe it's not working as well. They try another preparation. Maybe they don't form any antibodies to that one because it doesn't have the fragments or the changes or the defects, so to speak.
Would you say that we can determine that by a bioassay against IGF-1 levels, whether it's losing its effectiveness? If we're tracking, so if we do a simple bioassay where we basically administer a certain amount of growth hormone of a certain number of days and then go and get our serum IGF-1 levels. Now, clearly that's not the best tool in the world, but it's a practical tool for our tribe. Yeah. Would you say that that's a practical a way of measuring whether you're responsive to the growth hormone still, because you will see a decrease in IGF-1 levels? Is that valid?
You want to have your diet relatively in place, because remember, nutritional status is important. So
the same diet at that time, same diet when you're doing the bioassay. Yeah.
And it takes some time for IGF-1 levels to change. So they'll stay elevated for weeks and weeks, and they slow down. The binding proteins are in part there to slow the deterioration of the IGF-1. So only there's like at least there's there's more than three there's like 6 or 7 binding proteins.
Yeah, binding proteins, yeah.
Yeah, so those have various functions in different tissues, but part of that is
binding protein 3 is the one that we would be principally concerned about, I understand. Is that correct?
Yeah, that's the main one.
Yeah.
But there are others that have different functions. That's complex, and I've looked in that for a long time. I can't really speak too well on that, but it does take some time for IGF levels 1 to change. They'll become elevated. It'll take a while for them to level off. Mm-hmm. So, for instance, someone might need to use a growth hormone with a constant diet and follow it for a month or two and see how the levels change or plateau off compared to a month or two, and then maybe 2 months in with the other preparation to see. But yeah, the bioassay, the thing that's also worth doing if you're going to do those is get a growth hormone measurement as well, just to make sure, like, 'cause a lot of people are using stuff that's not pharmaceutical grade. They're getting it from the black market, just to check and see to what extent they're getting actually real growth hormone. So yeah, that could be done without a doubt. That's the way a lot of people are really attuned to that. As to how much that IGF-1 is actually producing growth, contributing to growth, that's probably a variable thing too, I think, to be honest.
And it's fairly controversial too, I would say.
Yeah, it really is. It really is.
I understand. Cool. All right, Scott, we've been talking for like an hour, 20 minutes. I'm gonna keep talking for 5 hours if I don't, call into.
Well, we'll do it again if you like.
I was gonna say maybe what we could do is at your convenience, we can get you back on and we can talk about some different subjects and different threads. You're a tremendous resource for our community. I thank you so much for taking the time out to come and talk to us. So thank you very much. Maybe to be honest with you, I'm embarrassed to say I don't know what your website is. Maybe you could just tell us what your website is.
I just tell people Google Scott Stevenson and put bodybuilding in, and you can go to fortitudetraining.net or drdrscottstevenson.com. BYOBBcoach.com is for the book.
Buy a copy of that book, guys. It's a very valuable resource. Lots of good ideas and good reading. They're highly recommended. As I said earlier, Scott got my money. So highly recommended.
Thanks.